Hello everyone, today let us discuss about glycogenesis and glycogenolysis. First let us come to introduction. Glycogen is a homopolymer made up of repeated units of alpha-D glucose, and its molecule is linked to each other by alpha-1,4 glycosidic bond and alpha-1,6 glycosidic bond linked branches for every 8 to 12 residues. Excess of glucose is converted to glycogen in vertebrates. Glycogen is the main storage polysaccharide in humans, animals, and fungi. It is especially abundant in the liver and skeletal muscles. In hepatocytes, the glycogen is found in large granules in the cytoplasm. Glycogen granules are complex aggregates of glycogen and the enzymes that synthesize it and degrade it, as well as the machinery for regulating these enzymes. The biochemical breakdown of glycogen to glucose to meet the energy requirement of the body is called glycogenolysis, while the process of formation of glycogen from glucose molecules is called glycogenesis. So first, let us come to glycogenesis. Glycogenesis is the synthesis of glycogen from glucose. Glycogen is synthesized depending on the demand for glucose and ATP, that is energy. If both are present in relatively high amounts, then excess of insulin promotes the glucose conversion to glycogen for storage in liver and muscle cells. The reactions of glycogenesis or glycogen synthesis are: number 1, formation of glucose 6-phosphate. Glucose is converted to glucose 6-phosphate by the action of glucokinase or hexokinase. Glucose, they react with ATP to form glucose 6-phosphate and ADP. Second, formation of glucose 1-phosphate. Glucose 6-phosphate is converted into glucose 1-phosphate by the action of phosphoglucomutase passing through obligatory intermediates, that is glucose 1,6-bisphosphate. So glucose 6-phosphate, they are converted to glucose 1-phosphate. Third is conversion of UDP glucose from glucose 1-phosphate. Glucose 1-phosphate is converted into UDP glucose by the action of UDP glucose pyrophosphorylase. This is a key step in glycogen biosynthesis. Pyrophosphate is formed, which is later hydrolyzed by pyrophosphatase into 2-phosphate molecules. Here, glucose 1-phosphate, they react with UTP to form UDP glucose with the release of 2 inorganic phosphate. Now let us come to the fourth step, it is glycogen primer. Glycogen synthesis cannot start from scratch. It requires a primer. This primer is a preformed alpha-1,4 polyglucose chain or branch having at least 8 glucose residues. The protein glycogenin is both the primer on which new chains are assembled and the enzymes that catalyze their assembly. The first step in the synthesis of a new glycogen molecule is the transfer of a glucose residue from UDP glucose to the hydroxyl group of thyroxine of glycogenin, catalyzed by the protein's intrinsic glycosyl transferase activity. The Nescan chain is extended by the sequential addition of 7 more glucose residues, each derived from UDP glucose. The reactions are catalyzed by the chain extending activity of glycogenin. Now let us come to the fifth step, that is elongation of glycogen chain. Once a chain of 8 glucose molecule is formed, glycogen synthase binds to the growing glycogen chain and adds UDP glucose to the 4-hydroxyl group of the glycosyl residue on the non-reducing end of the glycogen chain, forming more alpha-1,4 bonds in the process. The overall equilibrium of the path from glucose 6-phosphate to lengthened glycogen greatly favors synthesis of glycogen. Number 6. Branching in glycogen. Branches are made by the glycogen branching enzyme, also called amylo 1,4,2,1,6 transglycosylase or glycosyl 4,6 transferase. The function of this enzyme is to break a fragment of glycosyl residue at the 1,4 linkage and attach them into another glucose molecule on the chain to form the branching point by alpha-1,6 glycosidic linkage. This results in more number of endpoints of UDP glucose to add further glucose residues. Thus, branching enzyme results in extensively branched last glycogen molecule. Defect in glycogen synthesis and glycogen degradation results in accumulation of glycogen inside a cell, which leads to glycogen storage disorders. One such genetic disease is glycogen storage disorder type 4, called as Anderson disease, caused by defective branching enzyme. Now let us come to regulation of glycogenesis. Glycogen synthesis is strictly monitored to regulate the blood glucose level. It is activated in well-fed state and suppressed in fasting. According to basis of regulation of metabolic process, the factors regulating glycogenesis are: number 1, availability of substrate. In well-fed state, when blood glucose level is high, glucose 6-phosphate, the substrate for UDP glucose, is also high. This allosterically increases glycogenesis. Second point, hormone. Glycogen synthase, the key enzyme of glycogenesis, exists in active, that is dephosphorylated, and inactive, that is phosphorylated form. Hormones like glucagon and epinephrine are diabetogenic, that is, they increase the blood glucose level. Thus, they antagonize glycogen synthesis, which is an effective way of reducing blood glucose level and storing it for further use. These hormones succeed in their function by a series of biochemical reactions which results in phosphorylation of glycogen synthase enzyme, rendering it inactive. Insulin is an anti-diabetic hormone. It lowers blood glucose level by stimulating uptake of glucose by muscle cells and glycogenesis in liver and muscles. Now the third point, calcium ions. Calcium ions or cyclic AMP act as secondary messengers. This is an example of negative control. The calcium ions activate phosphorylase kinase. This activates glycogen phosphorylase and inhibits glycogen synthase. Now let us come to glycogenolysis. Glycogenolysis is the breakdown of glycogen with n number of residues to glucose, 6-phosphate and glycogen with n-1 number of glucose residues. Glycogen branches are catabolized by the sequential removal of glucose monomers via phosphorylasis by the enzyme glycogen phosphorylase. Glycogenolysis takes place in the cells of the muscle and liver tissues in response to hormonal and neural signals. In particular, glycogenolysis plays an important role in the fight or flight response and the regulation of glucose levels in the blood. In the liver, glycogen is a food reserve for the maintenance of normal blood glucose levels during fast, between meals, and during high-intensity physical activity. In the muscle, glycogen is an energy source for muscular activity. Therefore, glycogen breakdown occurs during contraction and only in muscles involved in the activity. Now let us come to glycogen breakdown. In skeletal muscle and liver, the glucose units of the outer branches of glycogen enter the glycolytic pathway through the action of 3 enzymes: glycogen phosphorylase, glycogen debranching enzyme, and phosphoglucomutase. Glycogen phosphorylase catalyzes the reaction in which an alpha-1,4 glycosidic linkage between 2 glucose residues at a non-reducing end of glycogen undergo attack by inorganic phosphate removing the terminal glucose residue as alpha-D-glucose 1-phosphate. This phosphorylasis reaction is different from the hydrolysis of glycosidic bonds by amylase during intestinal degradation of dietary glycogen and starch. In phosphorylasis, some of the energy of the glycosidic bond is preserved in the formation of the phosphate ester glucose 1-phosphate. Pyridoxal phosphate is an essential cofactor in the glycogen phosphorylase reaction. Its phosphate group acts as a general acid catalyst promoting attack by inorganic phosphate on the glycosidic bond. Glycogen phosphorylase acts repetitively on the non-reducing end of glycogen branches till it reaches a point 4-glucose residue away from an alpha-1,6 branch point where its action stops. Number 2. Further degradation by glycogen phosphorylase can occur only after the debranching enzyme, formerly known as oligo-alpha-1,6 to alpha-1,4 glucan transferase, catalyzes two successive reactions that transfer branches. Once these branches are transferred and the glycosyl residue at the carbon number 6 is hydrolyzed, glycogen phosphorylase activity can continue. Third, glucose 1-phosphate, the end product of the glycogen phosphorylase reaction, is converted to glucose 6-phosphate by phosphoglucomutase, which catalyzes the reversible reaction. Now let us come to metabolic fate of glucose 1-phosphate in muscles and liver. Glucose 1-phosphate, the end product of the glycogen phosphorylase reaction, is converted to glucose 6-phosphate by phosphoglucomutase, which catalyzes the reversible reaction. In muscles, the glucose 6-phosphate formed from glycogen in skeletal muscle can enter glycolysis and serve as an energy source to support muscle contraction. In liver, glycogen breakdown serves a different purpose: to release glucose into the blood when the blood glucose level drops, as it does between meals. The phosphate group of glucose 6-phosphate is removed by the enzyme glucose 6-phosphatase, which is present in liver and kidney but not in other tissues. The free glucose exits the cell via GLUT2 facilitated diffusion channels in the hepatocyte membrane. Now let us come to regulation of glycogenolysis. Glycogen breakdown is under a fine control through covalent and/or allosteric modifications of some key proteins such as phosphorylase kinase, glycogen phosphorylase, and protein phosphatase. Glycogenolysis is also regulated hormonally in response to blood sugar levels by glucagon and insulin, and stimulated by epinephrine during the fight or flight response. First, let us come to allosteric regulation. Certain metabolites that allosterically regulate the activities of glycogen synthase and glycogen phosphorylase regulates glycogenolysis. Glucose 6-phosphate, glucose, and ATP allosterically inhibits glycogen phosphorylase, thus inhibiting glycogenolysis. Glycogenolysis is enhanced when glucose concentration and energy level is low in the body. Second, let us come to hormonal regulation. The hormones, through a complex series of reactions, bring about covalent modification, namely phosphorylation and dephosphorylation of enzyme proteins, which ultimately control glycogenolysis. Hormones like epinephrine, norepinephrine, and glucagon in the liver activates adenylate cyclase to increase the production of CAMP. The enzyme phosphodiesterase breaks down CAMP. The hormone insulin increases the phosphodiesterase activity in liver and lowers CAMP levels. The hormones epinephrine and glucagon bring about glycogenolysis by their action on glycogen phosphorylase through CAMP. Glycogen phosphorylase exists in two forms: phosphorylated the active form, and dephosphorylated, that is the inactive form. The CAMP activates CAMP-dependent protein kinase. Protein kinase phosphorylates inactive form of glycogen phosphorylase to its active form, which degrades glycogen. The enzyme protein phosphatase removes phosphate and inactivates phosphorylase kinase. Protein phosphatase can dephosphorylate and convert active glycogen phosphorylase to inactive form, thus inhibiting glycogenolysis. Third is regulation through calcium ions. Calcium ions binds to calmodulin, that is calcium modulating protein, and directly activates protein kinase without the involvement of CAMP-dependent protein kinase, thus activating glycogen phosphatase and increasing glycogenolysis. So now let us come to conclusion. Glycogenolysis is the biochemical breakdown of glycogen to glucose, whereas glycogenesis is the opposite, it is the formation of glycogen from glucose. Glycogenolysis takes place in the cells of muscle and liver tissues in response to hormonal and neural signals. In particular, glycogenolysis plays an important role in the adrenaline-induced fight or flight response and the regulation of glucose levels in the blood. The reverse process, glycogenesis, the formation of glycogen from glucose, occurs in liver and muscle cells when glucose and ATP are present in relatively high amounts. In the synthesis of glycogen, 1 ATP is required for every glucose unit incorporated into the polymeric branch structure of glycogen. The glucose in the form of glucose 6-phosphate is synthesized directly from glucose or as the end product of gluconeogenesis. Thus, glycogenesis and glycogenolysis are coordinated to maintain the blood glucose level in the body.